This is the lac operon experience. Let's start with the big picture.
DNA is not free-floating within the nucleus. It resides on chromosomes and is composed of many genes. One particular cluster of genes in prokaryotes like E. coli is the lac operon. It is made of a promoter, operator, lac-C, lac-Y, and lac-A genes. The promoter binds to RNA polymerase. Lac-O, or the operator, binds to the repressor when negative control is active. Lac-C produces beta-galactosidase, which is responsible for splitting lactose sugar. Lac-Y codes for the production of galactoside permease protein, which channels lactose into the cell through the membrane. Lac-A codes for the production of transacetylase protein, which is responsible for exporting sugars out of the cell when they become too abundant. Upstream of this operon is another gene called lac-I and its specific promoter. It is responsible for coding a protein called the repressor, which is involved in negative control. Not all genes are always expressed because protein production costs the cell lots of energy. Why use more energy than needed and die? So to limit this energy expenditure, the cell uses negative control. Lac-I codes for a protein called the repressor, which binds to the operator, it's around here, creating a blocking device. In this case, RNA polymerase can bind to the promoter in the beginning of the operon, but cannot get past this repressor and transcribe lac-Z, Y, and A genes. Therefore, no beta-galactosidase, galactoside permease, or transacetylase would be made. However, some genes are transcribed constitutively, or at all times. An example is lac-I. Thus, the repressor is always made. To turn this negative control off, a lactose sugar must be present. Lactose in this case is called the inducer because it induces the transcription of lac-Z, Y, and A. It does so by allosterically regulating the repressor, so that the repressor changes shape and cannot bind to the lac operon at the operator site. No repressor means nothing is blocking transcription of the lac-Z, Y, and A genes. If there is lactose and no glucose available, the cell needs these proteins to use the lactose for energy.
This is a demonstration of negative control. RNA polymerase binds to the promoter but meets resistance by the repressor. But when lactose comes, the repressor changes shape and transcription occurs.
Now let's take a look at positive control over the lac operon. First and foremost, the driving force behind positive control is the inverse relationship between extracellular glucose concentration and the concentration of intracellular CAMP molecules. Here you can see that CAMP is derived from ATP, the enzyme adenyl cyclase catalyzing the reaction. Adenyl cyclase removes two of the phosphate groups from ATP, and our product is CAMP. In this diagram, we see that extracellular glucose concentration is high, and thus our CAMP concentration is low. Consequently, the CAP protein is unable to bind to the binding site on the DNA, which yields infrequent transcription of the operon. Conversely, when glucose concentration is low, CAMP concentration is high, and the CAP protein will successfully bind to the binding site, triggering transcription. The CAP binding strengthens the promoter, which is fundamentally positive control.
When glucose levels are high, RNA polymerase transcribes the lac operon infrequently. When glucose levels are low, CAMP is produced by adenocyclase and binds to CAP. Together, they bind to the binding site, and RNA polymerase can efficiently transcribe the lac operon.
Let's take a look at the system when all the genes are functioning normally. E. coli cells need two proteins in order to use lactose. They are galactoside permease and beta-galactosidase. Galactoside permease is a membrane transport protein that imports lactose into the cell and is encoded by the gene LAC-Y. Beta-galactosidase breaks down lactose into glucose and galactose and is encoded by the gene LAC-Z. Glucose goes directly into glycolysis. Galactase goes to a mediator step first and then to glycolysis. This is a representation of the LAC-Z mutant. The gene for beta-galactosidase is not present, so beta-galactosidase is defective. Lactose can still enter the cell, but will not be broken down into its glucose and galactose subunits. This is a representation of the LAC-Y mutant. Galactoside permease is not present to bring lactose into the cell because the galactoside permease gene is defective. Lactose concentrations in these mutants are much higher than in normal cells.
When the cell is functioning normally, all the LAC cells genes on the LAC operon are transcribed, and lactose enters the cell via galactoside permease. Once inside the cell, beta-galactosidase splits the sugars apart, and glucose and galactose eventually filter through glycolysis. When the LAC-Z gene is not transcribed or mutated, then the lactose sugar can get into the cell. However, there is no beta-galactosidase to split it apart.
The transcription of the LAC operon may or may not occur if there are mutations in the system. For the first case, if the LAC-I gene is mutated, this prevents the operator from recognizing the repressor. RNA polymerase binds to the promoter, and transcription occurs constitutively with or without lactose. In the second case, when the repressor is mutated again, but in this case, the super repressor always binds to the operator even when lactose is present. The repressor will ignore lactose and remain bound to the operator. In the third case, when the operator is mutated, transcription occurs even if absence of inducer, because repressor doesn't recognize mutant operator. Therefore, RNA polymerase binds to the promoter, and the LAC operon genes are transcribed constitutively. In the last case, when the promoter is mutated, RNA polymerase does not recognize the mutant promoter. Therefore, RNA polymerase will fall off the promoter, and transcription of the LAC genes will not occur.
If there is a mutation in the LAC-I gene and the repressor is made but not functional, or there is a mutation in the operator and the repressor cannot bind to the operator, then RNA polymerase will transcribe DNA continuously.
What happens to the operon system when there are equal levels of lactose and glucose in the environment?
The system will favor glucose and not transcribe any of the lactose genes until it is placed in an environment where there is low enough glucose for the adenylyl cyclase to start producing CAMP.
Why can rising levels of CAMP be considered a starvation signal in E. coli?
High CAMP concentration signifies low glucose concentration. Glucose is used by the body during cellular respiration to extract ATP. Low concentration of glucose means low amount of energy, which leads to starvation.
What happens to the operon system when there are equal levels of lactose and glucose in the environment?
The system will favor glucose and not transcribe any of the lactose genes until it is placed in an environment where there is low enough glucose for the adenylyl cyclase to start producing CAMP.
Why can rising levels of CAMP be considered a starvation signal in E. coli?
High CAMP concentration signifies low glucose concentration. Glucose is used by the body during cellular respiration to extract ATP. Low concentration of glucose means low amount of energy, which leads to starvation.
How does gene expression change in your gut-dwelling bacteria after you eat a cookie versus drink a glass of milk?
Eating a cookie is intaking glucose, in which it would go directly into the glycolytic pathway. Drinking the milk, however, is primarily lactose, and such, it would be metabolized through the lac operon.
That concludes the lac operon experience.